For my Module 4 video, I decided to do DNA replication in prokaryotes. There are three main steps in the replication of prokaryotes. The first is initiation, elongation, and termination, but we are mainly going to be focusing on initiation and elongation today. Initiation first occurs when proteins bind to the DNA, more specifically the origin of replication, which in this case is the red star. Helicase, which is the green square, then separates the two strands of DNA, almost looking like an unzipper action, creating two replication forks as a result. As the DNA becomes unwound into two single strands, as you can see in this picture, single-stranded binding proteins, which are purple circles, attach themselves to each of the DNA strands in an effort to stabilize them so that the rest of replication can occur and the strands do not reattach. In addition, gyrase, which is the orange boomerang shape, attaches itself to the DNA. The role of gyrase is to prevent supercoiling, as it is a subgroup of the topoisomerase. Next, we move into the elongation period, and primase comes into the picture. Primase is the light blue circle and always attaches to helicase. Primase acts on RNA primers, which are the dark blue rectangles, synthesizing them. Primase is essential to the process as it makes it possible for DNA polymerase III to do its job. The job of DNA polymerase III is to get the actual replication done. This is represented through the light brown arrows. It moves in the 3 to 5 direction, synthesizing the new DNA in the opposite direction, 5 to 3, due to the anti-parallel quality of DNA. Next, DNA polymerase III adds nucleotides to the strands. Primers must keep being placed along the strands in order for new DNA to be built. Because each primer is made of RNA, DNA polymerase I's job is to replace these RNA primers with actual DNA instead. However, there are sections in between each primer that result, called Okazaki fragments. Ligase's job is responsible for filling in the spaces in between each fragment. In addition, we must note that the bottom strand is the leading strand and the top is the lagging. The reason being that the bottom strand is synthesizing new DNA in the 5' to 3' direction. The top strand contains Okazaki fragments as primers keep being placed and are synthesized in the opposite direction. To answer the question in the prompt, in essence, DNA polymerase I's job is to replace the RNA primers with DNA during replication. If it were absent or no longer able to perform its function in the 5 to 3 direction, then RNA would not be replaced with DNA along the new strands, causing replication to be inaccurate and cease due to the non-functionality of DNA polymerase I. That is how DNA replication in karyotes is done, and here are my sources. Thank you.